Good morning, dear second year students. Today we are going to discussing about gene concept. History about gene concept. Gene was coined by W. Johnson in 1909. Yeah, God, 1908 proposed one gene, one product hypothesis. Gene theory was proposed by T.H. Morgan in 1911. L. Pauling and Ingram 1949 established the role of genes in protein synthesis. This is the history about gene concept. And next we are discussing about classical concept of gene. Introduced by Schuton 1902 and was elaborated by Morgan in 1930, Bates 1923, Muller 1927 and others, which outlined as follows. Genes are discrete particles inherited in Mendelian fashion that occupies a definite locus in the chromosome and responsible for expression of specific phenotypic characters. Gene concept, a year and scientist gene concept. Who scientist invented the new concept in particular year? In 1866, Gregor Mendel was proposed to a unit factor that controls specific phenotypic trait. And next, in 1902, Sir and Garrod one gene, one metabolic block theory. And next, in 1940, Badel and Tatum was proposed on one gene, one enzyme theory. In 1957, Ingram one gene, one polypeptide theory. In 1960s, and C. Enoshki and co-workers proposed to gene is a unit of recombination. G.W. Beadle and E.L. Tatum in 1948 proposed one gene, one gene hypothesis. The fine structure of a gene was proposed by Seymour and Benjor in 1962. S.K.S.D in 1965 proposed one gene cistron, one polypeptide hypothesis. Gene concept was given by Schuton. Structural genes. Genes are different types. So first we are going to discussing about structural genes. It regulates to produce specific mRNA. mRNA means messenger RNA. Determine the kind of protein to be synthesized. Operator genes. These genes act as switches to turn on or turn off the activities of structural genes. The structural genes, depending on operator genes. The operator genes involved in the switches to turn on or turn off. And next, regulating the elongation and termination of polypeptide chains. The operator genes regulates the two process, elongation and termination of polypeptide chains. And next, regulator genes. These genes produce certain proteinaceous substances called repressors, which prevent the operator genes from their action. Modifying gene. The gene cannot produce a character by itself but interacts with other to produce a modified effect is called modifier genes. Then the gene cannot produce character by itself. Modifying means changes. The gene which cannot produce a character by itself but interacts with other to produce modified effect is called modifier gene. Inhibitory gene. The next gene is called inhibitory gene. The gene which suppresses or inhibits the expression of another gene is called inhibitory gene. Suppress or inhibits means stop. Stop means stop. So the inhibitory gene involving in the suppress or inhibits the expressions of another gene. So this type of genes is called as inhibitory gene. This is the chromosomes. The genes present or come out from the chromosomes. Classical definition of genes. Gene is the unit of function. One gene specifies one character. The one gene specifies only one character. Recombination and mutations also involving in the gene chromosomes. This is another structure. We will like this structure. We observe chromosomes. This is the chromosome. This is a gene. Essential features determines the physical as well as physiological characters in genes. Situated in the chromosome, occupies a specific position known as locus, arranged in single linear order, occur in functional states called alleles. Some have more than two alleles known as multiple alleles. Alleles means the alleles present in genes. This works on genetic materials. Terms related to gene. Recon. It is the smallest unit of DNA capable of undergoing crossing over and recombination. Mutant. It is the smallest unit of DNA which can undergo mutation. The first one is crossing over and recombination, and second one is can undergo mutation. Cistron. It is the unit of function. It is the gene in real sense capable of synthesizing a polypeptide chain of an enzyme. Complan. It is the unit of complementations. And next, genes as unit of transmissions or cistron. The part of DNA specifying a single polypeptide chain is termed as cistron. The part of DNA specifying a single polypeptide chain is termed as cistron. A cistron can have 100 nucleotide pairs. In length, 30,000 nucleotide pairs. It transmits characters from one generation to other as unit of transmission. So this is a called transmission of cistron. Next we are discussing about genes as a unit of recombination or recon. The smallest segment of DNA capable of being separated and exchanged with other chromosome is called recon. A recon consisting of not more than two pairs of nucleotides. Gene as a unit of mutation or mutant changes. Mutant is the smallest unit of genetic material which when changed or mutated produce a phenotypic trait. Mutant is delimited to a single nucleotide. Genes in protein synthesis. Genes required for the protein synthesis and also genes essential for the protein synthesis. Jacob and Monod proposed operon model to explain the mechanism of gene action. Protein synthesis is regulated by three specific genes located on chromosomes: structural genes, operator genes, and regulator genes. These three types of genes involved in the protein synthesis. Thank you.